Hello all, I'm coming to you from beautiful Boulder, Colorado, and I'll be talking about disorder and entropy in molecular crystals. I'd like to thank the organizers for giving me the opportunity to present to you all today. So acknowledgements, this work is primarily done by Nate Abraham, who now, as of about 6 months ago, works at AbbVie, and Eric Dybeck, who's been working for several years at Pfizer. Funding from primarily from NSF, external collaborators, Graham Day and Dave McMahon at the University of Southampton. Transparency and Conflicts, I consult for Silicon Therapeutics on binding for entry calculations, and I'm a member of the Open Force Field Initiative funded by a consortium of pharma companies. So as we all know, that's why we're in this session, the predicting crystal polymer stability is important for drug formulation. If you don't get the right crystal form, you have the problem, the lowest free energy crystal form, you have potential problems in formulation as different forms come up, or hopefully not after it's already on the market. So it's very important to be able to predict what will happen for given molecules. We'd love to do it even before these molecules are synthesized or before they're actually put into large-scale production. So crystal structure prediction is becoming more accurate and accessible. The 6-blind prediction challenge, then the top groups generally place the molecules in the top 5 predictions, often in the getting the exact right experimental structure. Most other groups found polymorphs and ranked the true polymorph pretty high. So this is getting more and more, people are getting more and more capable of doing this. It still can be very computationally expensive. However, it's being used by major drug companies right now, as you all know, as cloud computing gets cheaper, and as we understand the underlying difficulties with and limitations of CSPs and energy functions. We know how to work around the problem. So standard CSP methods typically compare stability based on lattice energies. You find all the different packings and make the approximation that the entropy will cancel out between polymorphs, and you can rank it just based on lattice energies. But what we see is that generally you get far too many structures. If you're lucky, it looks like the left, where the experimental structure is the one predicted with lowest energy, but many times it's like the situation in the middle, where maybe you got it ranked highly, or more often than not, your experimental structure is nowhere near the lowest energy. So this is something we want to try to understand a little better. How much does entropic considerations play into this? Are these different minima actually are all part, or many of them part of the same free energy minima that they'd coalesce if you treated it properly? So yes, the current CSPs generally neglect entropy, assume that the entropic contribution cancels out between molecules. And so our research questions are really, is this T delta S contribution to the stability negligible? Is a quasi-harmonic approach to the entropy good enough? And what really is the structural ensemble of these crystals? So the systems, we've examined a sort of standard set of systems that have a lot of good experimental data and that we've characterized quite well. And they've studied them throughout several different papers. And what we do, we use sort of the brute force approach. Given a force field, and we have to stick with cheap force fields in order to do this, just cheap pairwise potentials, we're using OPLSAA, is we heat all the polymorphs off from low temperature to high temperature, and then we combine the simulations using multi-state reweighting to get a smooth free energy as a function of temperature for each polymorph. Now, this works quite well. The problem is, in order to find the free energy difference between two polymorphs at least one temperature, you need to compute the free energy difference between the polymorphs. So we generally use this modified pseudo-supercritical path, basically through an Einstein crystal, where you restrain the polymorph and you're calculating the free energy along the entire path. And then turn off the interactions, do an analytical correction between the polymorphs, and then expand it back. Turn on the interactions, release the restraints, and then that gives us a free energy at a single temperature. And then we can combine that with our free energies as a function of temperature to find the free energy difference at any temperature. Because we're doing the full statistical mechanics here, we can easily back calculate out the full entropy difference at a given temperature and the enthalpy difference as well. So what we find is that a quasi-harmonic approximation is actually pretty good for most small rigid systems with very little error, less than 0.1 kcals per mole. However, once you start looking at more flexible systems, and these are not even that flexible, these are just moderately flexible, you start having errors that are much bigger, up to half a kcal per mole, especially for some systems that happen to have a fair amount of disorder in them. And so, so there is a difference. You do have contributions that are important. Looking at this more quantitatively, T delta S can be pretty significant at room temperature. Looking at looking at polymorph differences, and even the enthalpy difference is temperature dependent. A harmonic approximation assumes that it does not the enthalpy does not change as a function of temperature, but we find definite changes that are not necessarily predictable at all. They're not directly related to polymorph flexibility. Now, of course, the important thing is, because since most people can do at least a harmonic approximation, if not a quasi-harmonic approximation, how much of a difference in the contribution to entropy is there? We find that they're reasonable for rigid molecules, that the harmonic approximation of the entropy really is pretty good. But for more flexible and disordered molecules, it's less and less accurate. So trying to understand sort of the reasons for these differences, one thing we looked at was anisotropic expansion. It turns out now that doesn't really contribute very much. If you look at the difference between MD compared to isotropic and anisotropic corrections, it really doesn't make that big a difference. Although there are some very interesting things here where you see an enthalpy compensation. You can have up to 0.4 kcals per mole difference in either the enthalpy or the enthalpy difference or the T delta S difference, depending on whether it's isotropic or anisotropic. So it's interesting to see that enthalpy compensation. You're sort of actually getting, accidentally getting the right free energies there. Not totally accidentally, but there's still some errors do it. But generally, anisotropic versus isotropic, it doesn't matter all that much. One interesting thing that did come up in this study is that the cell size is important. How big is your supercell? So what I'm graphing here is the difference between QHA using a unit cell and supercell. So all the comparisons I've shown so far between quasi-harmonic approximation are actually quasi-harmonic approximation with a supercell. If you look at the difference between quasi-harmonic approximation with a supercell and a unit cell, you actually get a decent amount of errors frequently, between 0.2 and 0.5 kcals per mole. Remember, this is not the difference in the entropy. It's the difference in the entropy difference, the free energy difference between polymorphs. And so there's a supercell dependent difference on the free energy difference between polymorphs. We have a pretty good idea where a lot of this comes from. Here I'm plotting these errors between the unit cell and the supercell against the change in the RMS in the box vectors. And you can see that the free energy difference between unit cell and supercell to a large extent has to do with the difference in box cell expansions. If you have a supercell, you'll have a larger expansion generally because of the higher entropy. And so this difference is actually not that dependent on whether the molecule is flexible or not. The filled in circles are the three most flexible molecules. And we can see that the cell size dependent difference in the free energy doesn't depend on whether it's flexible or not. So that's something I think that will need to be dealt with more, is that when one does harmonic approximations, you're still missing some of the entropy due to the cell size. So that's something to consider. How anharmonic is the potential energy surface of small molecules? To address this question, we heated the system, a supercell system, up to 300 Kelvin, and we're just interested in local rattling around. So we just carried out short 5 nanosecond simulations, quenched 100 structures down to 0 Kelvin, and then clustered the structures. This was done in collaboration with Graham Day's group at University of Southampton, and analyzed how many distinct clusters there are. If you heat it up and quench it down, how many different minima do you find that are really structurally distinguishable? So some molecules, it turns out, have very simple landscapes. heat them up to room temperature, cool them down, they go back to the exact same structure. Carbamazepine was an example of this, and the several other molecules are examples of this, especially the small rigid ones. But a lot of molecules do not. When you heat them up and then quench them back down, you get a lot of different structures that appear. For epsilon, for example, up to 100 Kelvin, it didn't really move. But once you got it up to 200 Kelvin, it found some completely different structures there. And at 300 Kelvin, you find that generally you quench down to a very large number of structures. All of those structures are part of the room temperature ensemble. And so these are all separate minima. And so the union of these minima is what's forming this configurational landscape. Tolbutamide has an even more complex temperature dependent landscape. If you look at that, so I look over the paper to look a little more at all the behavior you see. It's quite interesting. And overall, we see that small rigid molecules have a single minima, but flexible and highly symmetric, high symmetry molecules, which end up having a lot of disorder, have many. In fact, we just looked at 100 different structures, and for a lot of these, that's the number of distinct structures you get. Every single minima was structurally distinct. An important thing here is that we haven't entirely solved the CSP multiple structure problem, because most of the heterogeneity is between cells. It's because you have supercells, you have different conformations of equivalent molecules in different supercells. So this is actually a different problem that does need to be addressed, the fact that a lot of the entropy occurs between unit cells. And that, remember, it's not just that obviously, if you have more cells and they can move somewhat independently, you'll get larger entropy, but these are polymorph dependent entropy differences. Let me show you visualize some structural results from the tolbutamide case. So remember 100 minima, and then what we do here is look at the root mean square fluctuation of each of the atoms in each of the polymorphs. And you can see, for example, in form 2, red is more motion, blue is less motion. The whole thing is just rattling around back and forth equally for the entire molecule. But for something like form 4 or form 5, most of the molecule is locked in place and just the alkane tail is moving around. So it becomes clearer why there are entropy differences that are anharmonic that change from polymorph to polymorph. So we've come up with a new approach to entropy estimation, really to free energy stability estimation that includes all the entropy. And so we start out with the same temperature replica exchange simulations as with the full calculation that I showed earlier. But instead of doing the complicated pseudo supercritical path, turning everything off, we do something simpler. We take 20 or 30 configurations, minimize them with fixed Bach vectors, and then sum the harmonic free energies in a Boltzmann weighted way. So lower minima get larger weight. And then with those, that summed harmonics, averaged harmonic energies, we use that free energy difference to construct the difference between the two curves of the free energy as a function of temperature. So this captures the multiple minima visited by the MD and gets this sort of the size dependence as well. So see Nate Abraham's talk and 443C for more details on this. And so I'm going to spend a minute or two going over this slide, because I think this really captures a lot of the lessons that we've learned here. So first, with the gray dotted line, we have QHA independent of replica exchange MD. What does that mean? So we found that a lot of the error that occurs between MD and QHA is because QHA needs the right minima to match molecular dynamics. If you just minimize from an experimental structure, you don't necessarily find, because there's many minima, you don't necessarily find the minima that is actually consistent with the room temperature ensemble. You might end up in some subsidiary minima that doesn't vibrate quite the right way. So QHA dependent of replica exchange, what we do is heat it up and then gradually quench down to low temperatures to find a structure, and then do QHA with that structure. In that case, we can actually get pretty good agreement with MD MD in most cases. So although we have lots of anharmonic minima, if we find the right one, then they mostly cancel out to some extent. It's hard to tell how well this would work for more complex systems, but it's interesting that if you can find the right minima, which does require some heating and cooling, then you get a pretty good match to the free energies calculated with MD. So we do two other things here. We connect the two with QHA, and that's using the correct minima. And then also connecting the two free energies as a function of temperature with the multiple Boltzmann weights. And it works pretty well. And is only costs about 20% of what it would cost to do the full molecular dynamics corrections to the entropy. Down to point for the rigid molecules, 0.1, for the flexible molecules, 0.25, which should be enough to do a lot of screening. So some takeaway insights from this. So entropy cannot always be well predicted with QHA alone. It's not a big as effect as the force field, but it is not negligible. The differences between MD and QHA, it appears after doing this study, are due in large part to the difficulty in finding the true lattice minimum. If you just minimize for the experimental structure, you don't necessarily find that. Anisotropic expansion is useful for predicting good cell vectors, but not that significant for free energies. A system size, however, is quite important. If you're doing harmonic approximations with the wrong system size, there are moderate errors there. Small molecule crystals can contain many lattice energy minima within the same ambient temperature basin. These lattice minima usually involve the supercells. And finally, we have some nice techniques for estimating entropy that are cheaper that could be very useful for screening. So in the future, what we really need to do is apply this for more complicated molecules, especially hydrates, and perform these tests with better force fields. So thanks for listening, and contact me through the internets if you have any questions. Thanks.